Good afternoon, everyone, and welcome to the American Flora Endowment's Grow Pro webinar series. My name is Ping Yu. I am a graduate of Texas A&M University, where I received my PhD under Dr. Mengmeng Ge. I am now a postdoctoral research associate at University of Florida and a member of AFE's Young Professionals Council. My studies and research focus on weight management and biochar research. I am serving as the moderator for today's session on using biological and chemical control to manage whiteflies in greenhouses. On behalf of the endowment, I am excited to be a part of AFE's Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE's sponsors. This session is sponsored by BioWorks. BioWorks helps customers in the horticulture and specialty agriculture markets successfully deliver crops to market with biological-based solutions and support. If you'd like to see all of our sponsors and their sites, or if you are suppliers and interested in becoming a sponsor for a topic, you can find that information on AFE's website at endowment.org/growpro. Today's session has been pre-recorded in English. Dr. Lance Osborne will join us following the presentation to answer questions that you might have. We encourage you to submit your questions through the Q&A feature or the chat. We will answer as many of your questions live as we can before the end of the hour. Any unanswered questions may be answered through a separate email exchange following the presentation. This session is being recorded and will be shared to AFE's YouTube account. Through YouTube's accessibility features, you will be able to access closed captions in other languages. Without further ado, I'd like to present today's expert speaker, Dr. Lance Osborne. Lance Osborne is an entomologist with a passion for landscape and ornamental horticulture. Lance's research program focuses on the prevention and management of insect pests in nurseries and greenhouses. He is known as a pioneer in the development of biological control systems. Lance's involvement in integrated pest management trainings and work in the industry has led to numerous awards and notable recognition. After earning his doctorate in entomology from the University of California, Davis in 1981, Lance joined the Mid-Florida Research and Education Center, where he currently serves as Associate Center Director. Lance, welcome and thank you for presenting today on using biological and chemical controls to manage whiteflies in greenhouses.
Hello, I appreciate the opportunity to give a talk to the webinar series Grow Pro. I'm going to talk about using biological control and chemical controls to manage whiteflies in greenhouses. This slide shows a few of my co-authors that have been instrumental over the last few years in putting a number of these research projects into play and obtaining the data that I hope to present today. I'm going to talk about whiteflies. When I applied for this position at the University of Florida 40 years ago, I guess, they told me that I would never work on whiteflies again. Since the Bemisia tabaci invaded Florida in 1985, it seems like I've done little else than work on whiteflies. Prior to the invasion of the B biotype, we had the A biotype that was showing up in places in California and agronomic crops in the field, but it wasn't a pest of ornamentals. We didn't have a pest of ornamentals in the Bemisia group until 1985, when it showed up on hibiscus in a local greenhouse in Florida. It appeared to come via some tissue cultured plants, and then it spread throughout the state very rapidly, and then it spread throughout the country. Basically, we're calling some of the biotypes based on their geographic origin. So the B biotype is now being called the Middle Eastern Asia Minor, or MIM1, but I'm going to continue to use B biotype. In 2004, we had another biotype arrive, the Mediterranean, or MED, or as everybody else calls it, it's the Q-biotype. I'm going to refer to both of these as B and Q throughout my talk. Now, there are more than 40 proposed cryptic species. What a cryptic species is that it is a couple organisms that are distinct species, but you can't tell them apart with normal taxonomic visual cues. You have to basically use genetic and biochemical methods such as gel electrophoresis, PCR, in order to separate them and determine exactly what species it is. But it's nice from the standpoint that you can run these assays very rapidly. It doesn't take long to run quite a few through the process, and you can biotype them so you know if they're B or Q, which helps in tracking populations of whiteflies, and a very useful tool for looking at resistance spreading through populations of whiteflies. The Mesia tabaci group, again, which might be 40 species, attacks over 900 host plants. It vectors at least 111 plant viruses. Basically, B and Q are an example of resistance management, how we can try to manage resistance in a population of whiteflies that might occur in a greenhouse. I'm going to talk about management of these whiteflies as mainly in greenhouses, because that's what my expertise is. The foundation of a management program is scouting. Without scouting, you don't really know exactly what's going on in the population. It limits your ability to do biocontrol. If you spray, you need to be able to go out and have a hand lens and detect whether the pests are actually dying. So it's a foundation of any kind of efficient management program, and especially critical for IPM. There are three major goals of a successful whitefly management program. First is to help growers produce a high quality, marketable crop for the consumer. Implicit in that is that it has to be economical. They have to be able to have a crop that they can sell and actually make money. Okay, so that's number one. Two is to preserve the effectiveness of the chemical tools used to manage whiteflies. We don't have an infinite source of tools to manage these particular pests, so we need to preserve them. Three, we want to prevent the spread or distribution of difficult to control and possibly pesticide resistant populations. The idea in a management program is to start clean. You want your crops to start off on the right foot and have the best chance of going through the whole production cycle with the fewest pests or the fewest problems, okay, obviously. So if you're growing them yourself, you're going to have to work hard to keep your stock plants or your seedlings clean. And then, you know, if you have buy-in cuttings, you need to really establish a very good communication with your supplier. And you need to know and talk to them about their program of managing pests and other issues with the particular crop you're dealing with. You need to know if they're having any control issues with any of the other pests that might attack the plants, including whiteflies, mites, mealybugs, thrips, whatever. You need to know something about what they've been spraying on the plants. If you have a customer that doesn't want their plants that they're buying from you to ever be exposed to neonics, which some customers are dictating, you need to know whether they started somewhere in the chain with being treated with neonics. If you're intending to use biological control, you need to know what possible residues might be on that plant. We've had a number of complaints with people trying to start biological control on poinsettia, and the poinsettias have been sprayed with pyrethroids or some other compound that disrupts the biological control. If you're going to use biological control, you already need to have a source, a place where you're going to get your natural enemies and delivery schedules and everything already set up. You want to start before you ever see whiteflies, okay? So that you have these natural enemies that are actually acting like scouts in and of themselves. They're looking for the pest in order to survive, okay? So their lives depend on finding these pests in your crop. And if they're out there early, they have a greater chance of preventing uncontrolled population buildup. Okay, I said I was going to talk about chemical control. We've been working on chemical control of Bemisia ever since we found it in 1985. Pesticide resistance has been the driving force basically for all the research, including biological control, because we just use that as a tool to help manage resistance development. When we first found Bemisia in Orlando, Popka, Florida, really, the grower allowed us to come in and collect samples, and Dr. Leiby, who was here at the research center, he basically went through a process of developing data on just how tolerant or susceptible the population was to certain chemicals. And we found that the population that was in this particular grower's greenhouse was one of the most susceptible populations in the world to both organophosphate and pyrethroids. And so these tools worked for a little while in that particular greenhouse. Within 1 year, the population doing the same sort of studies had turned into being one of the most resistant populations to organophosphates, such as acephate, and pyrethroids, such as bifenthrin, that the grower, in order to grow a crop, had to dip the plants in a vat that had the maximum label rates of both these compounds. And basically, this grower wasn't killing them with toxicants. He was drowning them in this vat of pesticides. And this particularly worrisome if you looked at the PPE that these poor guys are wearing. They shouldn't be out there dressed like this when they're dealing with organophosphate or any sort of pesticides. The plants were being moved from this staging or dipping site over to where some ladies are actually putting sleeves on them and packaging them to be shipped out that day. So again, a total disaster. This isn't what we want to see. And this was the first sign that we had some major issues developing. We did a lot of work in trying to, later on when the Q whitefly was first detected, it came in 2004, 2005, and made its way around the country. We organized a large group of scientists from all over the country. We screened better than 44 products for efficacy on that particular biotype, or the Q-biotype, to try to develop and find tools that we could use to manage this particular pest that was known to be resistant to most of the other tools that we were currently using to manage the B biotype. At that point in time, with the help of AFE and Society of American Florists and ANLA, we developed an IR4, and we developed a management plan based on what we knew at that point in time about the effect of various pesticides on both the B and Q biotype. And we looked at trying to figure out what we could use at various stages of the production cycle of ornamental plants. This is version 3. We're up to version 7. We went to version 7, then we published one in 2017, which was hosted on the University of Florida website, where we still host our Bemisia management programs. From all the work of all the different chemicals that we evaluated, we finally decided, okay, we're coming up with these chemicals, but we need to know what kind of rotations can we put them in order to affect the most effective treatment or management plan. We published this in 2014. And again, it was a lot of people cooperating on this. We had Dr. Ron Otting, who was conducting most of the studies in cages in the hills of Georgia or mountains in order to prevent any kind of resistant whitefly from getting away, so that we could get some good data on managing the Q whitefly on poinsettias. We started with cages, and each cage had plants in it, and those plants were infested with 50 B-biotype whiteflies and 50 Q-biotype whiteflies. So basically, we had really a 50-50 population of B and Q at the start of the experiment. Obviously, when you're collecting whiteflies, it's difficult to get it 100% accurate, right straight down the middle, 50-50. But we were able to get a good distribution here, so that at the start of the experiment, we had equal populations relatively of the different biotypes. We had various rotations of different compounds, all containing neonics. And so we sprayed every other week or so. I can't remember exactly. I think it was every couple of weeks we would hit them with one of the 3 materials that we're rotating in. And we would, each week when we'd sample, we'd bring out a few whiteflies and basically biotype them to see what biotype was predominant in the cage and if we're getting any changes. Now, as it goes to Q, that means that the resistance profile of the population of whitefly in that cage is moving toward either a B or a Q or staying in the center. These are the results at week 8 of our biotyping. You can see that the water check basically stayed pretty much 50-50. Some rotations drove the populations very quickly to 100% Q, or it was building resistance into the population of whiteflies in each cage. Some were a little bit better at preventing both a complete movement toward Q. But the bottom line is that the more you apply pesticides, the greater the chance of building up resistance and having a Q population of Bemisia developing or taking over. The take-home message in all of this, though, is that all tested rotational programs manage the total populations, but none dropped to the population completely to zero. So we had all of these were basically working fairly well, and we have that paper on the website, so we can show you which ones work better than others. But rotations did work. Using less active materials for resistance management can still result in an effective overall program. So each compound that you use doesn't have to hit a home run. You can deal with compounds that may only have 70% effective control of the population, as long as it's rotating with other compounds. And the biotype Q is obviously less sensitive to certain products than the B biotype. If we have a contamination of B biotype in our colonies, we spray with distance, for example, and we, as one tool to basically drive the population toward being 100% Q. Now, there are a lot of new pesticides that have come out over the last few years that aren't considered neonics, with the exception of Expire. And so we've tried to put and develop data on the effectiveness of these materials. There are others, but we don't have data for those currently. We've had some issues trying to get companies there don't seem to be that interested in testing their materials on a Q whitefly, so we haven't put their data into these tables because we just don't have it. Okay, I'm going to talk about 2 different types of projects today, experiments that we've been running. We've ran one on compatibility, and basically this was using the maximum label rate of the pesticides that you'll see in the table to control the Q-biotype. And we also introduced into the study the predatory mite, Amblysia swirsky. So we're looking at the compatibility of the maximum labeled rate of the pesticides in the tables to control both the Q and the impact on the beneficial that we release to control the Q whitefly, the predatory mite, which eats thrips and whiteflies and some other mites. Treatment, then we had rotational trials where we had 3 different non-neonicotinoid pesticides that we would rotate, sort of like, every, we'd make a treatment and 3 weeks later we would make a treatment with a different compound. 3 weeks later, we would make another treatment. So we had 3 compounds that we rotated over a 9-week period, basically. And then we would, the salvia, we'd have 4 plants in one of, we had 6 cages per treatment. We had 4 plants in each cage. We would infest the cages with 100 Q whitefly 3 times at weekly intervals, which basically is 20 whiteflies per plant. And a treatment cages with Swirsky mite were inoculated with 20 mites per plant 3 times in a row, starting 1 week after whitefly infestations prior to the first insecticide application. So they've been put in for 3 weeks to establish, and there are whiteflies in there for them to feed on, so they wouldn't starve to death. So we would basically look to see at the end of this experiment, which they were much quicker. They're like 8 weeks or so to go from start to finish, depending on the health of the control plants that weren't treated. If they died, we basically stopped the whole experiment. So pre-treatment sampling to determine initial count of arthropods and whitefly biotype confirmation. So we take some out and make sure that we still had Q, was made prior to the application of insecticides. Treatment evaluations were made at weekly intervals, okay, just prior to treating with the next rotation chemical. We'd sample them and we would basically, prior to making the next treatment, we would look at, pull 5 leaves randomly from each cage. The adult whitefly counts were taken by the leaf sample method of turning the leaf over, counting all the whiteflies on that particular leaf, remove that leaf, bring it into the lab, count. If it was a compatibility study, we'd count the number of mites on the leaf. And if it was an immature whiteflies, or if it was just the rotation study, we would count all the immature whiteflies, including eggs. Okay, I'm sorry that may not be very clear, but we will be handing out a new program, and it will have the experiment detailed in it. Okay. So the test products for when we're looking at the compatibility with Swirsky, we did use a neonic. We used Mainspring, Rycar, Altus, Ventigra, Expire, and then we just threw Endeavor in to see what that would do. So they're all different classes. Okay. There were some drench, some foliar sprays. This is the percent control of med whitefly. You can see that there's a range anywhere from the 39 to 50% that didn't work very well on with Endeavor, to fairly high levels of control, 97 with Safari, which is the standard. But you can see with Mainspring, we got 90 up to 96, Rycar up to 95, Ventigra 94. Expire was fairly effective up to 89%. You can see, so all of them were judged to be effective on controlling the Q-biotype, okay? Whereas Endeavor wasn't. You can see that all but the Expire were compatible with Swirsky, which meant we always had populations in the cage of the predatory mite. They're feeding on the whitefly eggs. The only exception was Expire, and it's probably the Spinetta ram that was causing the population to be die out. In this we looked at the new insecticide efficacy rotation trials on the potted salvia with moderate adult whitefly infestation at low labeled rates. So we were trying to see, okay, could we make it more economical for the growers to use these and still get good control? You can see some of these initially didn't work. We've got low rates of success or control at 35. And then we added a second one that by then with these two, we ended up with 92.9% control. And again, at this stage, we had 97% control. And then for the overall mean of all the seasons, all the applications throughout the whole season, we basically had some that worked very well. So we have some rotations that worked very well that are non-neonics, and they also are at the lower rates, the minimum rate on the label. We feel that if you were to go up to the maximum rate, you could, these are 3 weeks apart. So if you were to make treatments at say 2 weeks apart, would be something to look at to see if we tighten this up a little bit, if we could get better control, because 3 weeks allows the population to build up to fairly easily between these applications. Okay, and this one is basically the same thing, except we started out with fairly high adult whitefly population. So at the end, there were a few breakthroughs where we didn't get very good control, especially the oil. Oil application every 3 weeks is just not going to cut it as far as managing whitefly. If it was, again, 2 weeks or a couple treatments right in a row, it might be much more effective. But again, we still have, if you look at the end of the season and the percent control, so we're talking 9-10 weeks, we do have some good rotations that offered us fairly high levels of control of this particular pest. So the bottom line is this: we have new tools that can be added to the tools we're using that were neonics. We have tools that aren't neonics that can be rotated and still get a high level of control. So that's all good news. We're not, so if you have customers that say you just can't use neonics, you do have tools to put together a management program that will allow you to grow clean plants and be able to ship them. Now, the new version has been under review, and it was basically approved last night with some minor changes in grammar and that sort of thing. And this will be posted on my website, or I'll give a copy to a American Floral Endowment, and they can put it anywhere, utilize it any way that they would like to. Okay, so after all of this, you have a fairly large list of pesticides that are known to be effective on the Q-biotype. You can identify all the pesticides if in your plan, if you're trying to make a management plan just for yourself and your crop based on what you want to do. You can identify all the pesticides registered for that particular crop, okay? You need to determine plant safety, no matter how safe it is under our conditions. We've had growers complain that, just right around the corner here, that they followed the same plan that I put together, and they end up with phytotoxicity because they have different water. They may spray the second week with a fungicide that could interact with the pesticide. So you really have too much to lose to not run your own phytotoxicity trial under your conditions in your nursery. Okay. You need to determine the label frequency. Each label has a resistance management statement on it. They tell you how frequently you should be able to use it on a particular crop or on a particular acreage. So you need to figure out how that will impact your management plan. You need to determine other use restrictions. So as you go down this list, you're going to find that you have fewer and fewer tools to use on the particular crop you're trying to protect. So you determine other use restrictions, such as the site. If the label says it's only for greenhouse and you're growing your plants outside, that product is not legal to use. So site can be an issue. You need to know if the plants that you're growing, if you have concerns with them being exposed to bees or other pollinators at some point in their production cycle or when they're being used. So you may have to cut a few products out, such as neonics, in order to protect the bee populations. And then customer demands. Again, we're starting to hear more people talking about they're told not to use any neonics in the whole life cycle of certain crops. Then you organize your treatment. So you're going to have a list of things that are known to be effective, ones that you've determined to be safe, ones that you know how many times you might be able to use it in the cycle of a particular crop, ones that fit that you can legally use or fit your restrictions. And then you put them all together and try to go from one MOA to the next MOA and not use the same compound like 3 times in a row, because then you can't use it anymore in the whole life cycle of the crop. So you try to manage. I recommend rotating between MOAs. And then you have to realize that in between these applications of insecticides, you may need to put another insecticide or miticide in there, or a fungicide or something else. So you need to take that in, other pests into account. Bottom line, though, is 100% reliance on pesticides equals resistance. So we're going to talk a bit about biocontrol. There are a lot of biocontrol programs out there. There's you can buy predators and parasites to manage the Bemisia whitefly. There's Eretmoceras. There are no Incarsias that really work that well. Some people are saying that certainly Incarsia formosa do. I've not seen it, at least in the heat and humidity in Florida. We haven't seen much luck with Incarsia formosa, but there are some people that say that that works. There's Eretmoceras, and then we in Florida have indigenous ones, but the only way we can get them to work is to use like banker plants, which we won't go into in this particular talk. You also will run into insect pathogens, insect pathogenic fungi. There are a couple, 3 or 4 different products out there. There's the Bavaria, the Botanigard, which we use as we compare everything to. It's one of the better products. Then there's Isaria formosa rosea, which I own the patent to, but it has some issues, but it is a very effective pathogen on whiteflies as well. In this slide, this is from a greenhouse at Gainesville, a student greenhouse where they grow poinsettias every year to sell. We had a section that we put under biocontrol, and then one section that was basically all chemical control. The red bars are the chemical control. We didn't start applying pesticides until week 3. And then we had to make multiple applications of Marathon and Flagship. And then everything was treated with Millstop at week 10. The biological control was basically always lower than the chemical control until week 10 and 11. At both treatments, we had plants that the students could sell. I think if we would have started with used one of the more effective pesticides here in the biocontrol section, we could have had clean plants and it would have equaled the control. But the bottom line is all the plants were sellable. And so it worked almost as well as the chemical control. Amblyssea swirsky, mite that kills whiteflies and thrips. Now this is the one we mentioned the compatibility work on. It is, we've had very good success with this predator on a lot of different pests, excuse me. We're using sachets. Sachet contains grain, a grain mite. This is acting on its own. Excuse me. There's a little hole here. The predators come out over a few weeks. And again, a very effective biological control agents for chili thrips and whitefly. The problem is that there are some crops that are watered overhead. These sachets, at least the ones we've used, didn't handle that water very well. And so we had to find another system to get them into the crop and maintain a population of Swirsky where we needed them. We came up with using banker plants. We've looked at almost 200 different varieties of peppers. Ornamental peppers came out as being one of the better ones because they put on a lot of flowers and they have what's called tuftomatia, which are these hairs that are in the axils of the leaf where predators like to hide. They live in those tufts and they can survive in there without having any prey on the plants or flowers for a period of time. When the flowers start to come onto the plant, the growth of the predatory mites is almost exponential. You can see predatory mites here on the hairs of the plants. This is a plant that was in a commercial greenhouse that was being protected using biocontrol. And this was one of the key components. It's a pepper plant with flowers. And we'd move this plant around every day. And the predators would crawl off and move into the crop. We've harvested some plants the other day, and the average was like 800 or so predators per plant. And they just keep reproducing. And they are immune, or we don't have problems where we have overhead water. Okay, this is a predatory mite, Swirsky. And if you look right in this one little area, the predator is pushing a whitefly egg back and forth as it's feeding on it. In this slide, this is a predator, and that's a tuftomatia there that was just shown. It'll go back in there in a second where there's another a mite that it doesn't want to feed on, and it runs away and immediately attacks this whitefly crawler. So it hits the crawlers, it hits the eggs, and then it will also attack the larger instar scales of the whitefly. Okay, again, this is Swirsky. This is a predator that we're very excited about. It's Delphastus pallidus, not Pusillus or Catalini. This is a new species that we've detected when we're working on all the other species of invasive whiteflies that we're dealing with here in Florida. This one seems to show up in almost all the populations, irrespective of what species. It's a very effective predator of whiteflies. We have a featured creature on this, published on the University of Florida. Again, something that we can get to you if there's a desire. These are the larval stages feeding on the scales. These can be used as a rescue. They will drop the population of whiteflies down very, very quickly. Here's the beetle. It's a little tiny red beetle. It's about half the size of Delphastus Pusillus or Catalini, the traditional one that we actually found, and at most of the populations that are being sold now came from stock colonies that we released here at the University of Florida. Again, wherever they are, they'll leave a little yellow dot that shows that the beetles have been there feeding. This is from a banker plant system that we use to introduce them into a crop. And again, it only takes a couple plants. This is the larval stage that feeds on whatever stages of the whitefly it can catch. Again, you can see these are the pupal stages of the beetle. This is the beetle. This is the Catalini. And you can see that they're much different in size. For some reason, this little one is tapping on its cousin, trying to probably mate. I'm not sure, but again, nothing's going to happen. Okay, quality control and efficacy. This is something that's absolutely critical when you're talking about managing Pamesia and being able to tell whether things are being effective or not. We've developed a system that we're trying to get growers to implement where we use lima beans from the grocery store. You can buy commercial varieties, but limas work best for us. As soon as they germinate and you can top them, you cut the growing point out. So you just have a plant with 2 large leaves. Okay, first thing you do is infest the plants. Now you don't want to be keeping, probably crop plants that are infested around. But what we do is we just put this in a crop that is infested. The whiteflies, or if you're using want to test this on mites, you can use it for mites or aphids or any other type of pest. So in this case, whiteflies are landing on the leaves, they're laying eggs, they're establishing a population. We age them in a greenhouse. We will put them in a cage to prevent them from getting out or causing any problems. if we're testing biological controls, we will take these plants out of the cage. We'll put a few biological controls on each leaf and come back in a few days to see if they're laying eggs and whether they're establishing. So it gives us an idea of the quality and the health of the biological control agents. We could release erythematosus on them and come back in a few weeks to see whether or not those parasites actually established and did anything on Bemisia. We can also take these plants and just dip them in the pesticides that you're applying to your crop. And in the flow of spraying your crop, we recommend to just spray some into a small container so that you could dip the plants directly into it. So these plants don't have any residues on them. They can be caged if you don't want to have the pests in the greenhouse, even though that's where you get them initially from. And it allows you to pay very close attention, and you get thorough coverage by dipping the plants. In this case, we're taking plants, we're getting ready to dip it into a fungal preparation. We have some issues with some of the fungal products that are being sold. No matter how good, excuse me, they are when they leave the manufacturer, if they're shipped in a big brown truck and that big brown truck stops to eat lunch or dawdles and you're the last delivery of the day, those materials can be exposed to very high temperatures. And there's times where we plate the fungus out and they're not up to snuff. In some cases, we've had completely dead product. So we recommend that growers test their material to make sure that they're effective. We dip it in the material, we let it sit like this, and then come back in a week or so to see whether or not they're doing what is promised of them. This is ambient relative humidity and everything in the greenhouse, the conditions that your crop is being grown at. We also recommend putting 1 or 2 plants in a plastic bag, so you can see the plastic bag is tied around. And then we open them up and we evaluate. Did you get the results that meet your requirements or expectations? So you can do it with pesticides for mites, for aphids, for whiteflies. We generally will tag or mark 1 leaf, and we'll count it before we dip it, and then we count it afterwards and look to see if we get the results we want. Okay, in this case, every whitefly on the plant was dead from fungus. And with that, I want to thank you for your attention. I know this is very rapid. I'm sure that I lost some people because I talked a little fast. I really appreciate it. And are there any questions?
Lance, thank you so much for your presentation. This is extremely informative. Now I'd like to open up to the session for Q&A now. If you have any questions, this is your time to ask away. We have a question from the Q&A feature. Somebody asked the biological control treatment, said banker plant. Can you elaborate on any successful banker plant systems for whitefly control in greenhouses?
Okay, we've had, like I mentioned a couple times, a couple banker plant systems that have been successful. One is the use of the ornamental peppers. We like explosive ember or red missile, a couple different varieties that put on lots of flowers, and they have the domatia that I mentioned. And this works very well for 2 species of predatory mite, Lamonicus and Swirsky, both of which have been touted as being good biological control agents for whiteflies. The other system that we use, and we've used it for years, and that's where the first Delphastus came from a banker plant or a papaya plant infested with a very host-specific whitefly called the papaya whitefly. And we can take a papaya plant with papaya whitefly and put it in a greenhouse with poinsettia. And as long as you don't put the plant right over the poinsettia, you don't have any problems. If you put it right over the poinsettia, it'll grow sooty mold from the honeydew that comes off of the papaya. So you have to put it over in a corner. And basically, the predatory beetles that we use and some parasites will feed on the papaya whitefly and then move out into the crop and attack Bemisia. And like we said earlier, you try to get these into the greenhouse even before you put your crop in. So it's already has a layer of biological control agents established in the greenhouse prior to even bringing your crop in there.
Okay, thank you. What is a cryptical species? Can you explain that?
Okay, cryptic species. We're seeing this happen now in both Bemisia and in the chili thrips group. There are actually like almost 40 different species that are basically classed under the Bemisia tabaci. We can't tell them apart using microscopes or traditional taxonomic tools. And the only way that we can separate them between the various species or groups that they belong to is to take them and crush them up. We can crush an individual whitefly, then we'll do PCR and gel electrophoresis, and basically we can tell whether that whitefly was a B or a Q. These populations are considered different species, but we just can't tell them apart any other way than doing that.
Right, thank you. Any other questions in the audience? Don't be shy. Now it's your time to ask away if you have any questions. Okay, we have another one here. How can you mix biological controls products with synthetic pesticides successfully? Do you have can you elaborate that?
Okay, we did touch on that a little bit when we talked about the efficacy and the compatibility studies. Okay, so you need to realize that just about anything can cause problems, even water. Okay, they're fragile, you can drown them and that sort of thing. But we do test these chemicals against some of the most important biological control agents. There are websites where you can go in and type in the agent that you want to use, say Swirsky, and then you can go through and type in the pesticide that you're most interested in, and it will come up to tell you whether or not the material is safe on Swirsky, how long its residual activity is, and all sorts of things like that. I know that BioBest and Copert both have these types of pages that are apps on the iPhone. So you can go to the iPhone store, type in side effects, and then you will get two things that come up. One will say BioBest side effects, and the other will say Copert side effects guide. And then both of those will work to help you figure out potential problems, okay? But you still have to realize that, if you spray in really high pressure, good coverage, you're going to impact the population. So nothing is 100% safe, but in general, you don't get 100% coverage, and you can spot treat different areas with compounds, but some have 8 weeks residual activity and can cause you problems for a significant amount of time, which are like 7 and bifenthrin or Talstar and those types of things. Okay. I would always ask where you get your beneficials from, to talk to the people about their particular cultures and their colonies and what they think about the safety of pesticides on their own on the products they're selling.
Okay, thank you. And here as a question, how does the cost of effective management of Bemisia using Swirsky compare to using parasitic wasp like Eremacus?
I don't live in the real world, okay? So I don't have to, I don't have the economics, and I haven't worked with an economist to really figure it out, okay? So the way we basically recommend that people do it is that, you figure out what your cost of pesticides are. And a lot of companies don't have that information. Once they do, they sticker shock, okay? But the bottom line is, whoa, I just lost you. Are you still there? Okay. The bottom line is, you know, you need to, it's both can be expensive. Okay. There are studies out there that show the chemicals are cheaper. There are also studies out there that show that biological control can be cheaper. We like the banker plant system because you can maintain colonies of on your own. You can rear the beneficials if you want to. And like the pepper plants, you just grow peppers and you inoculate them with the predators, and you can do it from a colony that you maintain even in your warehouse. And it isn't that expensive. Okay.
Yeah.
Not a good answer, but I don't have a good handle on it.
So we have another question here. What is the effect sulfoxyphor about Swarovski mite in poinsettia?
What is the effect or the
sulfoxyphor?
Okay.
I'm not sure if I pronounced it right.
The issue there is that there are, at least as far as I know, there isn't a compound that is that particular active ingredient by itself. To get that active ingredient, you have to buy expire, which is combined with the spinetoram, which probably is what is impacting the mite. Okay, so there's some questions of just how long expire is going to last, because there's some EPA is questioning its use already. And it was held up for a long time. They released it, and now we're starting to get rumblings that they're looking at it again. So I'm not sure how long that particular compound is going to stay on the market.
Okay, thank you. And we have another question. How about using botanicals as bioworks, MOTX?
We haven't tested that particular one. Okay, some of the botanicals are very effective. Okay, but we haven't had those products donated to us so we can evaluate their impact on the Q-biotype. We've been going through the classic chemistry and those types of things, and so there's only so much time in the day that we can test all of these things. I have no problem testing it. It's just we haven't had opportunity to.
Okay, now we have time for the last question here. What is the length of life cycle of Delphistus pallidus?
Oh boy, I haven't read the paper in quite a while. So I mean, they seem to live quite a while. I mean, they build up very rapidly. But I have one of my colleagues at Homestead published a paper on it. I'm actually an author, but I don't remember the details. We can have that posted, and I have a whole list of things that I want to put on the website that you can gain access to, like the chemical rotations, links to the side effects and some of those things, and papers on efficacy trials. But right now, off the top of my head, I can't remember.
Okay, thank you. So I want to thank you for joining us today for another session of AFE's GrowPro webinar series. We encourage you to view our full calendar at endowment.org/GrowPro. While you're there, check out our past webinar recordings, other grower-related resources, and research reports available to you free thanks to industry support. Our next webinar will be managing impatiens downy mildew with variety selection and production practices on September 14th at 1 p.m. Eastern. We ask that you please complete the brief survey about today's session. Your feedback will help us continue to improve the monthly webinars and provide additional topics of importance. Thank you for joining us today and have a great day. Thank you.